Let's now talk about intracellular receptors. These are also called nuclear receptor, which is somewhat confusing. And in reality, these receptors are located in the cytoplasm and only migrate to the nucleus when activated. So in general, they contain a C-terminal ligand binding region, a DNA binding domain, and an N-terminal domain that basically activates um, the function, the action, and generally serves to recruit a co-activator. Um, if you remember from biochemistry what zinc fingers are, in general, the DNA binding domain is composed by zinc fingers. So types of receptors that work that way are the estrogen receptor um, and also the thyroid hormone receptors. So once again, what we have is that in the free state, the estrogen receptor contains a binding site. Um, upon binding, a terminal helix is recruited and closes up. That in turn causes dimerization and exposes the binding site for the co-activator, which is the AF2 protein. Binding of the co-activator causes the transcription factor to be fully active, so we are now calling about nuclear transcription factor, which migrates to the nucleus, binds to DNA, and activates transcription. So um, we have seen several types of receptors. In general, they all act the same way. They all bind to a messenger. That binding to the messenger results in a signal transduction. And so we have that in general, both agonists and antagonists can be used as drugs. So for agonists, what we'll do is that we'll try to mimic the natural ligand for each receptor. In this example, we have a neurotransmitter, and normally the neurotransmitter will bind to the receptor by doing that, basically utilizing several different regions. So here we have a hydrogen bonding binding region, a van der Waals binding region, and ionic binding region. So when the uh, neurotransmitter is in place, we make both interactions with the hydrophobic portion of the receptor through the aromatic ring. We make hydrogen bonds to this serine here through the alcohol on the neurotransmitter. And then again, we have an ionic bond between the charged amine and the carboxylate. Binding simultaneously to these three regions causes a rearrangement of the receptor, then changes conformation. So when we try to design an agonist, we take inspiration from our hypothetical neurotransmitter. And then what we do is we alter its structure subtly, like for example, we exchange an amine for the alcohol, or we form a cyclic compound to stabilize this conformation, or we change the aromatic ring for another hydrophobic group here. And the idea is that the agonist will bind to the same pocket as the natural ligand. The important thing is that the agonist should follow the same stereochemistry of the natural ligand, because all these interactions tend to be stereospecific and so will only happen for one of the two possible enantiomers. And so here we see that, for example, in our hypothetical neurotransmitter, only one of the enantiomer binds, if in a normal way, and so in a way that leads to induced change of conformation of the receptor, because it contains three interactions. When we exchange the chirality, and so we take an enantiomer, what we have is only two of the interactions are possible. The third one, the highly specific hydrogen bonding interaction is not possible. We also have allostatic modulators. Allostatic modulators bind to another site, which is not the active, the normal binding site for the receptor. And that also causes a change in the conformation of the receptor. And in this case, this ligand can be used to activate the same receptor that recognizes the binding site, and is used basically to treat thyroid problems. So let's now talk about antagonists. So antagonists generally bind at the same site than the messenger, but do not result in activating of the receptor. In this case, thus the antagonist has different interactions, and that prevents the changing conformation of the receptor. So antagonists are also called blockers because they prevent binding of the chemical messenger, so they act in opposition to the natural messenger. So reversible competitive agonists have affinity but no efficacy on their target receptors. And increasing concentrations of the messenger will actually end up displacing the antagonist. So how do we design antagonists? So if we look again at our hypothetical neurotransmitter, um, these are the three interactions that we discussed. And basically this binding changes the conformation of the receptor, by induced fit, and basically that's the conformational change that results in cell signaling. So antagonists in general bind to the same binding site, and in fact they typically bind better with higher affinity. What they do is that occupy additional portions of the receptor. In this case, we have added a hydrophobic group that makes contact with another hydrophobic region in the binding pocket. And this different mode of binding then results either in different changing conformation of the protein, or in other cases can prevent the conformational change in the receptor. And so we don't have cell signaling. Okay, another class of antagonists are irreversible antagonists, just like what we saw for irreversible inhibitors of the enzyme. Upon binding, we have a chemical step that results in a covalent binding, and basically irreversible inactivation of the receptor in this case. Some example of irreversible antagonists are the propylbenzylcholine mustard, which is an analog of benzylcholine mustard, and basically they work on the acetylcholine receptor. What they do is upon binding to the acetylcholine receptor, they react with nucleophilic sites on the enzyme, and so here we have chlorines that are good leaving groups, resulting in irreversible binding and irreversible inactivation. We can also have allostatic antagonism, and that basically reflects what we've seen with enzymes. So we have a change in the receptor conformation that then prevents binding to the natural ligands. Another type, or a subtype rather, of allostatic inhibitors are those that statically interfere with binding to the messenger site. So binding of the antagonist blocks primarily to basically a neighboring binding site, blocks the binding site for the messenger by sterics. And so the antagonist competes once again with the messenger, not directly, but through this allostatic inhibition. And so increased concentration of messenger do not displace the antagonist. Another peculiar category that we have with receptors and we didn't have with enzymes is the concept of partial agonists. So those are agents that act as agonists but produce a weaker effect than a natural agonist. And so there are generally different mechanisms involved in this. So either the agent binds but does not produce the complete induced fit reorganization that is necessary for binding site, or the agent can bind in different modes, one of which activates the receptor and the other one which results in antagonism. Or in the case of receptors that are present as in different subtypes, the partial agonists may bind, but not completely to one of the receptors, but not to all of them. And so the partial agonists bind at a given receptor, but differ in the amplitude of the functional response. So although they are agonists, they actually compete with the agonists, and so they look like competitive antagonists in the presence of a full agonist. So they're typically used in the case of receptors that can cause addiction very easily. So for example, we'll cover the use of partial agonists in the case of treatments of opiate dependency. So these partial agonists bind to the, for example, to the opioid receptor with a weak morphine-like activity, so that results in much weaker effect of euphoria and analgesia. However, they prevent binding of the, of drug of abuse, for example, in the treatment of dependency, and so therefore they're given as a preventive measure to addicts to basically discourage their use of drug of abuse. We then have another type of binders, the so-called inverse agonists. So inverse agonists bind to the receptor binding sites and cause a different change in conformation. The receptor is not activated, and the normal messenger is actually blocked from binding to the binding site. So for that reason, they look like antagonists because the natural ligand cannot bind and result in activation. So they block basically the inherent activity at the receptor. So, how do these drugs affect the receptors? And I think this illustration gives a simple idea of the various possibilities. So in general, a receptor in the resting state contains a mixture of inactive conformations and active conformations. And so what the agonist does is basically facilitates, shifts the equilibrium towards the active conformation. And additional agonists will do the same, and so stabilize the active conformation. Antagonists, on the other hand, stabilize the inactive conformation of the receptor by various mechanisms. Inverse agonists prevent binding of the natural ligand, and so basically stop the natural activity of the resting state of the receptor. And on the other hand, partial agonists activate the receptor, stabilize the active conformation by but they don't switch the equilibrium to the right by as much as an agonist.